Hi. First off, I would like to say thank you for the opportunity to present our work on microbiomes in freshwater quality assessment at the first DNAQUA International Conference. Freshwater ecosystems are inhabited by a diverse range of organisms that live above and below the water's surface and range in size from too large to pick up to the microscopic. In order to obtain and maintain a water quality status of good or higher, we make regular quality assessments based on biological quality elements, such as invertebrates, macrophytes, and fish. One large and diverse group of organisms present in freshwater ecosystems that is currently not used for assessment is the microbiome, as it cannot be observed with the naked eye. Through DNA sequencing, it is now possible to access this population, and we have worked to explore the question, can the microbiome also function as a biological quality element? As we transition towards DNA-based assessment methods, the assessment data we collect evolve alongside our methodology. Rather than collecting specific groups of organisms, we now target short gene fragments from conserved regions in organisms, such as the mitochondrial and ribosomal DNA, in order to collect data with greater ease, accessibility, and exponentially increasing scope. The DNA-based data we collect is not always directly translatable into the conventional protocols. For example, not all organisms have suitable reference sequences present in the available databases, and we also collect data on organisms that are not included in the conventional protocols because they cannot be distinguished by the naked eye. And it is in this data that we also find an opportunity. DNA-based water quality assessment holds the potential for the inclusion of the data that is not compatible with the conventional protocols in order to find potential new indicators and even entirely new quality elements. The microbiome is an example of a large and diverse group that has now become accessible. And microbiomes are already known as strong indicators in other environments such as technical ecosystems, thus they are worth exploring. In order to put our studies into the microbiome into perspective, we need to start at the beginning. Our initial study into the use of DNA-based assessment methods started with the analysis of 51 Danish freshwater streams to explore if we could use metabarcoding of the invertebrate populations as an alternative to the conventional index focused on invertebrates. The sequencing data we obtained from this study showed that we were able to capture known indicator organisms, as well as a number of other invertebrates that are not included in the conventional index. In addition, we found that while metabarcoding is not quantitative, that the relative abundance of individual groups was present on a gradient. For example, some organisms were observed more often in ecosystems of a lower ecological status, whereas others were observed more often in those of a high ecological status. And these observations correlated quite well with the indicator's values in the conventional protocols. The primary result of this study was that DNA sequencing of the invertebrate composition is suitable to separate streams by the quality categories defined by the Danish Stream Fauna Index. And this study was published in Ecological Indicators last year. Now that we knew that targeting a stringent population for analysis was suitable for estimating water quality, we wanted to broaden our perspective and explore if we could include the microbiome in these analyses. One primary reason for this is that microbes are the most abundant colonizers of any ecosystem, and they are known to be environmentally sensitive. In addition, available reference databases and information for microbiomes exceeds that of any other taxonomic group by 10 up to 100 times. The potential for microbial indicators has previously been shown in, for example, contaminated soils and technical ecosystems. And the large microbial diversity present in any ecosystem provides a great opportunity for finding potential new indicators for ecological quality assessment. To explore the potential of including the microbiome in freshwater quality assessments, we applied a broad-range metabarcoding approach to the freshwater streams from our invertebrate study. This approach captures all three domains of life, bacteria, archaea, and eukaryotes at once, and one primary result of this study was that especially the bacterial and eukaryote composition is able to separate the individual streams on a gradient based on the ecological status that we knew the streams to have. We achieved our approach by targeting the 16 and 18S Rrna Operon, a conserved region that is present in all organisms with evolutionary differences. Some regions in these genes are so conserved that it is possible to design barcodes that are able to capture all three domains of life at once. This comes at the cost of lower resolution data, but broadens the scope of the populations that are able to be captured. And as all three domains are analyzed at once, they are more comparable than if you were to analyze these three domains individually. This study showed us that the complete data from all domains, as well as the individual domains, reflect the ecological status of the samples analyzed, and that correlations between the presence of individual groups were observed relative to ecological status. This shows that we are able to find potential indicators in all three domains of life. We also explored the potential of the sediment microbiome in relation to ecological quality assessment. We did this by performing microbiome analysis of 31 freshwater stream sediment samples using the protocols for the Earth Microbiome project. The sequencing data obtained from this study truly showed just how diverse sediments truly are. We obtained 10 to 100 times more microbial taxa per sample compared to other biological quality elements. Initially, we aimed to investigate which sediment layer was most suitable for water quality assessment studies. But interestingly, we observed that when comparing the samples across locations, that water quality or ecological status, as was determined by conventional protocols, was a stronger determinant for the microbial community composition compared to the sediment sampling depth. We used the upper 5 centimeters of sediment to explore the relationship between the microbiome composition and the ecological status of our 31 sampled streams. One major result of this analysis was that the 31 sediments could be separated relatively well by ecological status based on the microbial community composition. In addition, the individual microbial populations were observed in a similar fashion to the invertebrate populations from our initial study, where, for example, some microbial populations were observed more often in the higher ecological status categories, whereas others were observed more often in the lower ecological status categories. This suggests that the microbiome can report on ecological status. The final and most preliminary study we performed aimed to investigate to what degree the sediment microbiome is reflected in invertebrates living in the sediment and vice versa. To achieve this, We performed microbiome analysis of 140 invertebrates of interest in Danish freshwater quality assessment and compared them to our 31 sediments from Danish streams. Two major observations could be made. Part of the microbiome overlaps with that observed in the invertebrates. But at the same time, the invertebrates were also observed to have their own individual microbiome, as well as microbiota that are associated to different groups of invertebrates at different taxonomic levels. This highlights the potential for using the microbiome as an alternative and high-resolution quality element. In summary, our studies have shown us that in essence, the microbiome is connected to all elements of a given freshwater ecosystem. The microbiome in the sediment, and by proxy the water column, is reflected in the invertebrates inhabiting the ecosystem, and vice versa, and the same goes for the other biological quality elements. In essence, all individual elements, be it existing BQE, new microbiome data, or combinations thereof, are able to report on ecological status, and this provides great potential for improving the resolution and sensitivity of our assessment protocols. So to answer the question we started with, can microbiomes be used as biological quality elements? Yes, they can. Thank you for listening.